Microscopy, the light microscope. Light is used and is passed through the specimen and focused by lenses. A magnified image is produced by the objective lens, and this is magnified again by the eyepiece lens to give a real inverted magnified image. The number of times greater the image is compared to the object is the magnification. How clear the image is called the resolution. Light microscopes are limited in the amount of magnification they can produce because of their relatively poor resolution. If they produce images above 1500 to 2000 times greater, then the image may be blurred. During the 1930s, the electron microscope was developed, and this allowed scientists to see the ultrastructure of cells in much more detail. They could see the organelles more clearly and see the internal structures of these small structures. It also allowed them to see viruses. A beam of electrons focused by electromagnets rather than a beam of light focused by glass lenses is used. The wavelength of electrons is much shorter than that of light, so the electron beam can get in between small structures such as ribosomes and allow a clear picture to be seen even at great magnification. So, electron microscopes have high resolution and high magnification. As we can see, the transmission electron microscope focuses a beam of electrons through a thin section of material stained with metal salts. The beam passes through some parts of the section and hits a screen. Where electrons were absorbed by the specimen, the area on the image appears dark, and where the electrons pass through, the image appears light. A photograph of the image on the screen is called a photomicrograph. The scanning electron microscope directs a beam of electrons from above onto a prepared whole specimen. Depending on the contours of the specimen, the electrons will be scattered, and thus a 3D image of the specimen will be developed. Sometimes colour is added to give a false colour image. You've probably seen plant cells under the microscope. You may have noticed that they are usually larger than animal cells, such as the cheek cell, and have a cellulose cell wall around the outside. They also have a large vacuole in their centre and chloroplasts, organelles that contain chlorophyll where photosynthesis occurs. Differential centrifugation. Okay, the light and electron microscopes allowed scientists to see the structures inside cells. However, that alone didn't tell them what these structures did. In order to find out the functions of the different organelles that are in cells, we have to isolate them. This is done in 2 stages. Some leaf tissue, spinach leaves work well, is placed in a cold isotonic buffered solution. The cold temperature inhibits enzyme action, and the isotonic solution ensures that no water enters or leaves the organelles by osmosis. The buffer maintains a constant pH. The tissue is broken up using a mortar and pestle, or a blender can be used. This breaks up the cell walls and plasma membranes and releases the organelles. The resulting green mush is called the homogenate. This is filtered and then centrifuged. Ultracentrifugation spins the tubes of homogenate at a very high speed, increasing the force of gravity, as happens in a spin dryer, so that the organelles are pulled to the bottom of the tube. At slower speeds, the heaviest organelles, the nuclei, come down. If the supernatant fluid is now taken off and spun at a higher speed, smaller organelles such as chloroplasts and then mitochondria come down. By taking the supernatant liquid and spinning again at a higher speed, organelles such as ribosomes come down. Many investigations using isolated organelles have revealed the functions of the cell organelles. Studies with the electron microscope have shown us the structure of the organelles, and, as always in biology, we can see how the structure is related to its function. Let's look first at structures found in eukaryote cells, the cells of protoctists, fungi, animals, and plants. They all have a true nucleus, hence the name eukaryote, bounded by a double membrane or envelope that has pores in it. These pores allow molecules such as RNA to pass out of the nucleus. Inside the nucleus is a jelly-like nucleoplasm and chromatin made of DNA and histone proteins. Just before a cell divides, the chromatin condenses into chromosomes. The nucleolus is a small spherical body in the nucleus. It is here that ribosomes are assembled and RNA to be used during protein synthesis is made. Continuous with the nucleus is the rough endoplasmic reticulum, a system of membranes enclosing flattened sacs called cisternae. The system of membranes gives a very large surface area, and they are covered with ribosomes. Proteins are assembled at the ribosomes and then pass into the cisternae. From here, the proteins pass to the Golgi apparatus to be modified. Ribosomes are small organelles that are not bound by a membrane. In eukaryote cells, they are called ATS ribosomes. The S refers to a Svedberg unit and means that these are slightly larger than the ribosomes found in prokaryote cells. ATS ribosomes are about 25 nanometers in diameter. Each ribosome is made of ribosomal RNA and protein and consists of 2 subunits. It is here that proteins are assembled. Smooth endoplasmic reticulum, SER. Like rough endoplasmic reticulum, but without the ribosomes all over the surface, it may be more tubular and less flattened than RER. In the smooth endoplasmic reticulum, lipids are made, synthesized and stored. Carbohydrates are synthesized and stored. There may be lytic enzymes, enzymes that break down substances. Mitochondria. These are rod-shaped, about 1 to 10 micrometers long and bound by a double membrane or envelope. The inner membrane is highly folded into cristae to give a large surface area for attachment of the structures that are involved in the manufacture of ATP during aerobic respiration. The space between inner and outer membrane is also involved in this process. The fluid matrix of the mitochondria is where 2 other stages of aerobic respiration, the link reaction and Krebs cycle, take place. The most important function of mitochondria is to produce ATP. Cells that are very metabolically active, e.g. muscle and liver cells, and cells that are involved in active transport have many mitochondria because they need to produce large amounts of ATP. Mitochondria were probably once free-living bacteria that became incorporated inside the cell. They have their own DNA, naked and circular, like bacterial DNA, and divide each time the cell divides. All your mitochondria came from your mother, as the egg contains mitochondria. The mitochondria in sperm do not pass into the egg at fertilization. Mitochondrial DNA is used in studies of ancestry. Okay, now it's time to look at centrioles. These are found in some fungi and protoctists and in nearly all animal cells. They occur in pairs in a special area called a centrosome. Each is a hollow cylinder containing 9 sets of triplet microtubules with proteins holding them together. They produce the protein threads that form the spindle for nuclear division and that also make up the cytoskeleton of the cell. Plant cells do not have centrioles, but the spindle threads form from proteins in the cytoplasm. Cilia. The surface of some animal cells have cilia, which are fine hair-like structures. Each cilium is made of microtubules arranged in the same shape as centrioles. They may beat and move the whole organism, as in the case of single-celled organisms such as paramecium, or, in the case of cilia in cells lining the trachea, they may move a band of mucus in which particles and bacteria are trapped. Cilia also waft the released egg into an oviduct. Microvilli. Cells that are involved in absorption, such as cells lining the small intestine and some kidney tubules, have projections called microvilli. These increase the surface area for more efficient absorption. Chloroplasts. These organelles are only found in plant cells and in some Protoctis cells. They are disc-shaped and about 2 to 10 micrometers long. They also have a double membrane or envelope, and the inner membrane is highly folded into a series of stacked membranes, thylakoids, to give a large surface area. The chlorophyll pigments used in the first stage of photosynthesis are situated on the thylakoid membranes. The fluid-filled stroma contains the enzymes needed for the second stage of photosynthesis. Starch is also stored in chloroplasts. Chloroplasts were probably once free-living bacteria that became inextricably bound up with host cells and are now organelles that are essential for photosynthetic organisms. They have their own DNA, naked and circular DNA, like the DNA in bacteria. Cellulose cell wall and cellulose. Plant cells and some protoctists, algae, have a wall made from cellulose. This gives strength and prevents the cell from bursting when it takes in water. Fungi also have cell walls, but their walls contain chitin. Prokaryote cells. Remember the bacteria? These fellows have been around for a very long time. Indeed, for about 3,000 million years, they were the only life forms on the planet. They are everywhere, and there are many types of them. A few cause diseases, but most are very useful. They managed to live here for a long time with no other life forms, but all other life forms would perish if bacteria disappeared. They play a vital role in decomposing dead remains and recycling substances vital to living organisms. We, each of us, have more bacteria in our mouths than there are people on Earth, and we have even more in our gut, and we need them. Babies are born with their heads facing downwards so that they pick up bacteria from the mother's anus to colonize their gut. The diagram shows the structure of a bacterial cell. On the left side, there are structures that are always present, and on the right side are structures that only some bacteria have. Bacteria have prokaryote cells. There is no true nucleus, no nuclear envelope, and no membrane-bound organelles such as mitochondria, chloroplasts, RER, SER, or Golgi. The chromosome is made from naked DNA, so it has no proteins with it, and carries the genes. The wall is not made of cellulose, but of something called peptidoglycan. The cytoplasm is a jelly-like matrix in which are embedded other structures, as it is for eukaryote cells. Bacterial proteins are synthesized on the smaller 70S ribosomes. Some bacteria have a flagellum, or many flagella, for movement. Some bacteria move quite fast, and some move at the same speed as continental drift. Some have a slimy capsule. Many pathogens have a capsule, and it protects them from the immune response of their host. Some have plasmids, small loops of DNA that may have genes for antibiotic resistance. Some have inwardly folded membranes giving more surface area for photosynthesis or respiration, or a mesosome that may be involved with cell division and formation of new cell walls. Some have pili, hair-like structures that help attach them to host cells. Bacteria don't do mitosis. They have no linear chromosomes or apparatus to make a spindle. They divide by binary fission. The DNA replicates so the single chromosome is copied and, if they are present, the plasmids are copied. The cell then divides into two, with each new cell receiving a chromosome and a plasmid. Tissues. A collection of similar cells and any intercellular secretion produced by them that is specialized to perform a specific function is known as a tissue. The study of tissues is called histology. Tissues are organized into organs, and organs are organized into systems. Just before we finish this section, think about viruses. They are rather odd in that they cannot carry out any metabolism, and they have to hijack another organism's cells in order to make copies of themselves. They are hard to classify, as it is difficult to say that they are truly living. They are not cellular, as they have no membrane, no cytoplasm, and no organelles of any kind. They contain nucleic acid, but it is either RNA or DNA and not both. They are all very small and can only be seen with an electron microscope.